Hi, my name's Andy Pitman. I work for Plant and Food Research in New Zealand. I'm based in Lincoln in the South Island, and I'm predominantly working on the bacterial side of the disease complex in New Zealand. We have a particular interest in the impact of Liberobacter on seed tubers and then the subsequent production effects on potato production. We previously showed that unlike in the US, where you get very little sprouting of diseased tubers, that we got quite a high incidence of disease of sprouting in those tubers, up to 90%. And so that was a pilot experiment, and in the last year we've actually been looking further at that work, and we've set up a replicated trial in two locations, one in Lincoln, as I mentioned, in the South Island, and one in Pukekohe, which is in the North Island, where Liberobacter was first detected. The idea of those replicated trials was to actually start to look at, again, whether diseased tubers could sprout, whether they could produce growing crops, and what the impacts were on those growing crops of the Liberobacter in the seed tubers. As a result of the experiment, what we found initially was that, again, as in previous experiments, we had a high incidence of sprouting. So we had around, on average at the two sites, about 70% sprouting, which is completely different to what has been observed in the US. And of those sprouted tubers, many of those showed stunting initially, so growth over the first 7 weeks of those plants was reduced and suppressed. However, they appeared to catch up, and many of those plants produced marketable tubers at the end of the season. We actually, at flowering, what we did is we also took samples of leaf tissue to test for Liberobacter to see whether it was in the plant tissue, and then after we'd done that, we actually subjected the plants to psyllids, and those psyllids were considered to be clean of Liberobacter or Liberobacter negative, to see whether those psyllids had an effect on the plant as well as the Liberobacter from the seed inoculum. Now, overall, what we found at the end of the season when we looked at yields was that actually yield was dramatically reduced up to 40% in those from those tubers which had Liberobacter as seed inoculum. However, if you looked at those different treatments with the TPP, there was no effect on yield of the presence of those cold psyllids. What we also found when we actually took every single tuber from every single plant, and we also fried those. Well, we looked at specific gravity, and we found that the specific gravity overall was unaffected. We also fried those tubers and found that overall in our disease scoring system, which was a disease score of 0 to 10, that there was no significant effect on the tubers of having seed inoculum. However, what we did notice was that specific plants did produce what we would consider to be zebra chip symptoms. So out of the 600 plants that we tested, we may well have observed ZC symptoms in 1 or 2 of those plants. And we have also tested some of the tubers from some symptomatic plants to see whether Liberobacter was in some of the daughter tubers. And we did detect in some of those tubers, very, very rarely, we detected Liberobacter. So that's really a summary of the work that we've done over the next year, and over the following year we're going to be replicating that trial again to see whether the findings that we observed this year stand up.